We are pleased to welcome Dr. Anuswa Roy Chaudhary, Assistant Professor at IIT Bhubaneswar, to our conference. Dr. Roy Chaudhary's significant contribution to the field of molecular biophysics and biochemistry, particularly in understanding the mechanics of ATPases and their role in DNA replication, have earned her widespread recognition. Her academic journey, which includes prestigious fellowships such as postdoctoral fellowship at Harvard Medical School, showcases her dedication to scientific excellence. We are excited to have her share her insights with us today. Dr. Roy Chaudhary, we request you to proceed with your talk.
I have just one request to Dr. Donosua and Dr. next speaker, Dr. Subhasis. Both of you will be leaving after this talk. I think you have to go to airport or something. So I'll request to finish your talk sharp 20 minutes. And I have a bell here so that both of you can finish it and we should have a lunch at least by 2:15 to 2:20. I would like to Okay, go ahead, please.
Okay, thank you very much. First of all, I would like to convey my sincere thanks to Professor Kundu and the organizer for giving me this opportunity. So, Professor Kundu has asked me to present my work in a popular way. So, it's hardly we get chance to present our work in a popular way in a multidisciplinary, audience. So, it's a challenge for me too. So, stop me anywhere where it doesn't sound understandable. So, the topic that we have been discussing since morning, like science and society, those science and societies are very much interrelated. So science contributes to society, society influences science. How science contributes? By giving the research focus, knowledge transfer, etc. And society provides the important funding, policy, and regulation. By doing both, by collaborating these two, we see problem solving like healthcare and climate change, economic growth, new industries and jobs are created, entrepreneurship and new products and services are created, improve productivity, reduce production cost, modernization of existing sectors like education, manufacturing, agriculture, ease of learning, environmental sustainability, and all of them together help to take decision by the government, and finally nation building happens. So, science plays an important role in the society. So, what society provides to science? The complex problem, the practical problem. And science tests that. And by testing, they explore and discover. Then, community analysis takes place and finally benefits an outcome. Therefore, this is a very synergistic interrelation that has to be fostered. However, the journey of the science and society is not very smooth because there are many misconceptions between the societies. The science awareness is lacking, so we do not really know whether we should trust the scientific ideas. And sometimes we are also, as a scientist, are confused that whether we are able to fulfill the society's expectations. So, enhancement of public perception and science is the key. That is how we have to go. So, the science education and our awareness is very important. Science education prepares students for the future. It promotes the critical thinking. Education fosters creativity, promotes scientific literacy. Education is the important for the economy. So science education is important for nurturing the brains, which are budding as well as existing, who are contributing to the nation building projects. So that's why the concept of citizen science is very important in today's world, which encourage observation, questioning, planning, analyzing, and communicating among the communities. So scientists could be the catalyst for this citizen science concept. So that's how the different sectors of the science are related to the society, and interconnection is important. And among all the sector I have talked about, healthcare is one of the important sector. Winston Churchill told long time back that healthy citizens are the greatest asset any country can have. So let me go back to my work, where my third area is cancer and the gastrointestinal cancer. Why I'll talk about that, and precisely stomach, pancreas, and colorectal cancer. We work on cancer prevention, we work on cancer treatment. So first I'll focus on the prevention stuff. So we work on gut microbiome. You know that in our gut microbiota, there have trillions of microbes that are present, and several of the diseases are caused by their interrelation. So what happens is, in the gut, the balance is nicely maintained by good bacteria and the bad bacteria. However, when we are like having like lifestyle problem, we don't do exercise, we eat a lot of, fast food and all. And so what happens? Pizza Hut, Domino's effect, fix our system. Also, we have a lot of medication which are killing the good bacteria. So inside the gut, where the good and bad bacterial balance should be maintained, is hampered, and dysbiosis develops. So dysbiosis is the predisposition of pro-inflammation, then to chronic inflammation, leading to the severe disease like colorectal cancer, IBD, and several other things. So dysbiosis, see, if you check it out, this one, dysbiosis can cause a lot of different diseases, not only cancer. So what happens is, when the gut is inflamed, that can actually transmit the signal to the several different organs, to the gut lung, gut brain, gut heart, and many different organ axes. Thereby, many different diseases are caused by dysbiosis. So how to deal with that? Probiotics. Probiotics are the good bacteria. If you intake the probiotic, it can maintain the balance and thereby rectify the gut microbial balance. Therefore, probiotics are important molecules. However, each probiotics are strain specific. Therefore, detailed characterization is important. So these are the way we characterize the probiotics. It has to be non-pathogenic, short generation time, resistance to digestive enzymes, effective adhesion to gut lining, acid bile tolerance, sensitive to antibiotic, anti-genotoxic properties, lactic acid producer. If all of them are there, then we consider them as a good probiotic. And this is how we have been characterizing. So first, what we do when we get the strain, we do the whole genome sequence analysis, and then using bioinformatic, metabolomic, and functional studies, we characterize the probiotic strains. So I'll be talking about two such important probiotic strains that we have characterized, and side by so we are doing the in vitro assay, and side by side we are analyzing the whole genome sequence and see that how the molecular level we can get an insight, because in the probiotic field people are seeing the activity, but very less work is done in the molecular mechanism. So we have seen that both our strains are biofilm forming strains, which are important because this biofilm formation helps them to sustain in the gut nicely. They are also showing antioxidant properties important for the rectification in the gut inflammation. They are safe to use. We have used, in the colorectal cancer cell line, and we see that there is no toxicity. Antimicrobial properties, they have very nice antimicrobial property. And also, they are anti-biofilm effects. So one of the challenge in the, treatment, be it, like hospital isolates or any infection, is like biofilm forming pathogens. So MRSA is one of such pathogens giving a lot of trouble. So we see that these probiotics can inhibit biofilm formation by MRSA. With that, we wanted to figure it out what are the active components in the probiotic secreted molecules that are contributing to these activities. So we have analyzed the genome, and we found that bacteriocides are present. Not all probiotics have the active bacteriocides, but we see that in our genomic sequence. We predict the sequence of the bacteriocide, and further purification and characterization is going on. Therefore, these two strains appears as promising probiotic for their application to improve gut health. So that's our prevention part that we are working on. Next is cancer treatment part. So what we are basically doing is finding out a good target and inhibiting that target. Target means some proteins that are contributing to carcinogenesis. So if we can inhibit them, we can cure cancer. So gastrointestinal cancers we have chosen one of the most common cancer across the world. And 26% of the global cancer incident and 35% of all cancer-related deaths are in the worldwide is happening with the GI cancers. You can see the colorectal, stomach, and pancreas. These are important GI cancers that we are working on. In Odisha also and in India also, these are very prevalent cancer that needs to be addressed. So we are focusing on that. And so as I said that we are finding out a target. So AT82 appears to be one such target, which is an ATPase. And interestingly, it degrades ATP, and how that energy is, like what exactly they are doing is not known. They are involved. There are many cancer that is like AT82 is found to be overexpressed and contributes to the carcinogenesis. However, how it is doing the carcinogenesis is not known. So we have been working on the regulation and mechanism of AT82, as well as we wanted to, we have seen that whatever may be the mechanism, if we do, we so we are trying to find out the small molecule inhibitors as well as RNA-based inhibitor technology like microRNA, or whether that plays any role. So I'm going on. I hope I'm on time. How much time is left? Okay, so very fast. So what we have seen, you see that this is like early and the later stage of cancer. You see that AT82 is overexpressed. This is a biopsy samples from stomach cancer. Next, we have seen that in the stomach cancer cells, where you do see that AT82 is overexpressed in hypoxic condition, but in normal condition it is not. And side by side, we have performed that when AT82 is overexpressed, what is the consequence in the patient samples? We see that the high expression of AT82 decrease the survival of the stomach cancer patients. So it could be a promising detection marker in stomach cancer. Next, we wanted to find out, okay, this is AT82, but cancer doesn't happen for a single protein. So what is the interactome of the AT82? So computationally, we figured out the interactomes, and then we are working on the, that interactomes, and we see that with AT82, these are some of the important oncogenes are over. For stomach cancer. So we have knocked down AT82 and checked out the proliferation assay, and we did see that inhibition of AT82 reduced stomach cancer progression. We have also seen that inhibiting of AT82 could reduce metastasis in stomach cancer. We wanted to find out what is the regulation of this overexpression. So we have checked out, since the AT82 is overexpressed in hypoxic condition, we have checked out which is one of the regulator in hypoxia, and we see that HIF1 alpha activates AT82 for transcriptional activation to upregulate AT82 in stomach cancer. So basically what happens, like this is our model, that in the hypoxic condition, when HIF1 alpha is there, HIF1 alpha binds to AT82 promoter, activates AT82, so that AT82 is overexpressed and mediate cancer. Another important project that I'm just telling you, that microRNA 217 also reduces pancreatic cancer. This is done with the pancreatic cancer system. So microRNA 217 reduces pancreatic cancer progression by inhibiting AT82. So together we can say that HIF1 alpha activates AT82 expression, although in a different cancer, stomach cancer, and microRNA 217 inhibits AT82 expression. And in other group have shown that microRNA 217 also inhibits HIF1 alpha expression. So if we can inhibit, like if we apply microRNA 217, that could be a double-edged sword, which reduces both AT82 as well as HIF1 alpha. So that could be the something we are thinking about. And in addition to that, since AT82 appears as a good target, so we are trying to figure it out. The small molecule, we have done drug repurposing as well as synthesis. And this is like only the monomers. It's a huge protein. It's a hexamer, homohexamer, and each monomer has having three important domain: two ATPase domain, one bromo domain, and both ATPase domain and bromo domains are considered as popular cancer target. So some efforts have been initiated for the bromo domain, but nobody has addressed the ATPase domain. So we are working on that. And so we hope that with the small molecules, I have another story where we have seen that in chemotherapeutic drug, with the 8082 is showing synergistic role. So combination strategy could be a good way to go in order to deal with the 8082 mediated carcinogenesis in general. So this is an important slide of my students, collaborators, and special thanks to again Professor Kundu and the other organizer for giving me this opportunity. And I'm happy to address any question. Another slide I have that is important also. So I hope that science and the society will collaborate each other and we will have a better belief among ourselves. Thank you.
We extend our sincere thanks to Dr. A. Roy Chaudhary for her brilliant presentation. I would request Professor Gopal C. Gudu to please present the memento to Dr.
Quick question.
Yes, sir.
So it was good talk. So that very short question. You have shown that HIP1 alpha hypoxia-induced factor regulates ATAT2 expression in this gastric cancer. But what happens if you check it under hypoxic condition? That is number one. And number two,
we have done it in hypoxic condition only.
So number two, HIP1 alpha also regulates VEGF expression and angiogenesis.
Yes.
Have you explored that?
I did not explore, but in other cancer, people have shown that yes, ATD2 also comments the angiogenesis, but mechanism is not yet known. That is something like we can do it.
Thank you.
Thank you.
Okay. I request Dr. Anil Chatterjee to please present the memento to Dr. A. Roy Chaudhary. I'd also request Professor S.K. Acharya, CEO, ITBI Cell, Sophitorium, to please present the potted plant to Dr. A. Roy Chaudhary as a token of appreciation.
Not and. No, thank you.
Moving on with the last speaker before the lunch session. It is a great honor to welcome Dr. Subhasish Chattopadhyay, a distinguished scientist and educator from Niger, Bhubaneswar, to our conference today. Dr. Chattopadhyay is widely recognized for his pioneering work in cellular immunology with a focus on understanding immune mechanisms in infection, cancer, and inflammatory diseases. His impactful research has not only advanced the frontiers of biological sciences but also opened pathways for novel therapeutic approaches. Please join me in extending a warm welcome to Dr. Subhasish Chattopadhyay. So please proceed with your.
One small announcement just before Dr. Subhasish start his talk. Before everybody breaking for lunch, we like to have group photo, one with this background, INSA background, and another Nasi background, with all speakers and Sophitorium faculties and everybody. So some will be standing, some will be sitting, and take the photo, and then we'll go for lunch.
Hello. So very nice afternoon, and thank you all, especially the Sophitorium group, Dr. Padi, Dr. Misra, and the distinguished delegates, guests, and the teachers. Like I am also a teacher, but I am talking about our teachers and great leaders and advisors Professor Kundu and Dr. Anil Chatterjee. Because of them, we are here, and I am personally connected with Dr. Kundu, and in our native language, we call Gopalda for last 25 years. But after coming here in Bhubaneswar for last couple of years, I am more connected. I am looking for his advice, and in different forum, because of my awkward vocabulary, I described Gopalda as encyclopedia or mini encyclopedia in cancer biology. And Anilda, and I have to give a new name, that the most youngest mind in elderly age. I told in the morning, I am planning for different grants, that Anilda, I like to take your blood for my experiments to test how your T cells and macrophage are different, if any. Anyway, so the topic is about science and society. So today I thought this way, that I will not present any data slide, because I thought it is a group of science where non-biological or non-life science audience will be also there. So that's why we will not present any scientific data, but the essence will be biological sciences, especially immunology. Now, we started with this morning sessions about Sophitorium, and Dr. Naik and others understood and told that things. It is basically the GAN munch, right? The audience where they get Sophie, that is GAN. And if you think about our India, that innovations, product development, all these things were there, at least narrated, well narrated in our prehistorical time in Ramayana and Mahabharata. And also, if you think about where is the proof, and what the science we do is observable science, but there are greater science where we can feel but may not get the findings, that is difficult science. And there is a quote in terms of creations, like we all know the story of Big Bang, that from a single entity, from nothing to something, and from something to Big Bang, it happens. So we are all generated from the single dot, and that has been narrated, that I am the creator, I am that person. That is in Upanishad, especially the Brihandanak Upanishad, that they quote Aham Brahmasmi. That means I am the Brahman. In other word, in the modern time, I am the creator, and I means we. We are all created from one molecules or one dots actually. So we'll talk about our own defense mechanism, that if we defend us without attacking others, then we survive. And immune response and immunity is like that. So we are working on cell-mediated immunity, that is basically directed T lymphocytes. Our class 8 and 10 knowledge about blood lymphocytes, T lymphocytes are there. And this is my brief background. I'm not going to the details because we have to go faster. I'm also putting a timeline. So we are working, I am working in NISA since 2009, and my field is cellular immunology. And I have a couple of experience in postdocs in abroad in USA, and I am involved in regular classroom teaching. We are publishing different journals. So now, what the Nobel laureates? We talk about big people. Like in the morning, we heard about Elon Musk, right? But why are not talking about the recent, yesterday and day before yesterday, Mr. Gukesh, who got the award in 18 years, got in chess. That is also creativity. So we have to think about holistic approach, and we feel proud, and we have been proud, and we are being proud since the time started. The Indian culture and history is quite rich in productivity and creativity, and that's the other name of Aham Brahmasmi. So Carl Milston, all are Nobel laureates. If we start these things, that will be a seminar on what Nobel laureates are talking about science. And interestingly, they are in life science only. So I have just depicted only 3 Nobel laureates: Milston, Rolf Zinkenagel, and Jim Allison. By the way, Jim Allison got Nobel Prize recently in 2018. He's coming. His work on Gopalda knows that in cancer immunology, especially discovering the cancer checkpoint inhibitors. So all are, I'm not going to narrate their topics. You can read it. All are talking about the uncertainty coupled by creativity and prosperity and success and peace of mind for the society. So all are talking like product development is at most important, but to develop product you need another P, that is patience, perseverance and persistence. If we do not do basic science, where the product will come? First we should have the knowledge. Once we gather knowledge after long research, then only the product will come. It's not a machine. We are not going to the machine. I really respect AI and ML, machine learning, artificial intelligence. But before that, we should not be machine, and we should not be machine, and we should have our own intelligence before going to artificial intelligence. That's my personal concern. Please don't take it other way. The point is that we have to go with basic science. This is a famous quote of uncertainty principle. In physics, we have Heisenberg uncertainty principle. In immunology, there is another uncertainty principle by the Nobel laureate Rolf Zinkenagel. What is saying, anything and everything is possible in a contextual manner, but it should be, I add, should be repeated and very much contextual. If you are thinking for anti-disease product, it should be reproduced and it should be healthy. So what we need from bedside to bedside research, that is translational research. So we are working on cell-mediated immunity. This is our background work. I have worked on regulatory T cells. We are working on viral immunology in collaboration with Dr. Shoma Chattopadhyay in ILS. We are working on cancer immunology and experimental immune suppression, where without cancer we can induce immune suppression. Cancer induce immune suppression, but cancerous immunosuppressive drugs can also induce, and also basic immunology. Immune systems is very much relevant to the homeland security in the current time. The border securities, the terrorism, anti-terrorism and all these things, we have to know that things, we have to have a very good immunity. And immunity won't come with drug, vaccine or tablets or medicines. It will come through holistic approach of lifestyle. We the scientists, the biologists talk about big things, this protein level, lipid level, carbohydrate level, different sugar level. Are we thinking that we have to change our lifestyle first? Then all the medicine will work, because what it affects, it should have side effect. So we have to be aware that we should not take too much medicines rather than change the lifestyle. So these are the different type of avatars of T cells, where we talk about cell-mediated immunity. This is about Tregs in cancer, autoimmunity, and homeostatic conditions of normal immunity, where we worked on. This is about two coins, like two tails of a coin, two sides of the coin, head and tail. Autoimmunity versus cancer. In autoimmunity, you get excessive immune responses, inflammatory responses, which is deleterious. And in cancer, you lack immunity. What does it mean? What is the take-home message? Balance. We should to have balance in life. We should not be too much aggressive for anything. We should not be biased for anything, even for spirituality. If you see that things, those who are yogi, those who are highly in spiritual level, they have problem. And you have seen those terrorists or dacoits or all these things, they have also problem. It's just like autoimmunity versus cancer. So we have to have a holistic approach of life. Now, this is a crazy idea. We know that viruses induce cancer, but recent topics are coming up that the viruses can be utilized to combat against cancer. And this is the fact. And this is a crazy fact. It might be productive stupidity. And there are oncolytic viruses. It's an engineered viruses where they have tested that it can induce anti-cancer activities. And recently it came in the social media that COVID anti-cancer as anti-cancer virus. The question will be whether it will be really true. I will ask another question. What do you mean by true? What do you mean by truth? Truth is a relative word. What we see today, it will be changed tomorrow. Just a simple example for class 7 standards school in CBSE and state board. 10 years ago, there were 9 planets in the textbooks. There is only 8 planets now. What is true? Can you explain? It is the contextual truth. That is our things that we have to progress with the reality and contextual observations. So this is the things that by Dr. Ankit Bharat from Northwestern Universities published in Journal of Clinical Investigations that they are used COVID as anti-cancer. As I mentioned, COVID and antiviral and anti-tumor immunity are the reverse side of these things. We can think about for societal activities. And there are different types of immunotherapies for drugs, medicines, cytokines, all these things are involvement. And we are working on, this is our working model at this moment, viral immunology, cancer immunology and immune suppressions, and basic sterile immune activations and inflammatory responses. And we are working with immune activations of macrophage, immune modulation. And immune modulation, I was thinking with Dr. Chatterjee, that immune modulation is a very generalized things. Like you take XYZ drug and test on immune cells, it is activations or inflammations or immune suppression we can test, and that has a future potentials for different diseases. Chikungunya, I will not talk about because already Shoma has discussed about the introductions and different things. What I will tell that it is a multi-organs diseases. Simple viral infections by the female Aedes mosquito can be dangerous for us. And in India, it is coming up. And you can see that things. This is the latest scenario in 2024, I believe, 2019 onwards. And in Karnataka and in Maharashtra, there are still cases. This is the latest update. And this is the scenario in 2024 that what is the spread of chikungunya. And there are mortalities, but it is not about mortality. It is about the severity. As Shoma told, I have seen and we have seen that a chikungunya patient could be dangerous for future, for their lifestyle. And there are inflammatory cytokines. I'm not going to the details. The mouse is not, you can see these things. These are the inflammatory cytokines. We are working on that. And macrophage, we have found that is a resident host for after the viral infections. And we have worked on different aspects of chikungunya immunology. Like we have shown that during the infection, the macrophage undergo apoptosis, programmed cell death. And we have shown that apoptosis followed by the infections and inflammations could be regulated by HSP90. We have also worked with MAP kinase, what Shoma told, and Gopalda asked the questions. It is not about the macrophage inhibition. We have found the non-structural protein of the chikungunya, NSP2, actually physically interacting with the MAP kinases. That is our observations, and that has a future antiviral drug scopes. As I told, we have the leads from the bench side. We are waiting for the bedside. We have also worked with different ion channels. Recently, Nobel Prize to TRIP channels. David Julius got it. We have worked on TRPV1, that is here. And we have looked for different calcium channels. And also worked on TLR4. We all know, like with lighter sense, in serious sense, that all the viruses have certain receptors, entry point receptors, and that's the specificity. For example, a plant virus cannot infect us unless we engineered us to be plant. Similarly, a bird virus, cow virus, may not infect us because we may lack the receptor. For chikungunya, only 4 or 5 receptors were known, and we've also contributed in the society. We have shown the toll-like receptor, that is TLR4 here, you can see these things, is one of the co-receptor for the virus. And if we block the receptor, it not only restrict the infection but also reduce the viral severity and disease severity in animal model and also with human blood samples. This is the same thing what she told. We have contributed for the immunological part, and also we were also in the part of correspondence that the anti-hypertensive drug could be useful for anti-chikungunya infections. This is our basic immune activations without any virus. We have worked on T cells, we have worked on TRP ion channels, and we have also shown that during immune suppression, calcium influx is all known for any physiological conditions, and it is mostly reported for inflammation. We are the first, we have shown that it is also induced during immune suppression and also enhancing TRPV1. The mechanism details are awaited. COVID, as I mentioned, a crazy idea that it is known as proactive stupidity. Already in the beginning, I told that Ankit Bharat, not Biksit Bharat, Dr. Ankit Bharat has shown that COVID-19 can be beneficial towards cancer, to combat against cancer. Shomlata is here, our PhD student, and she is also a topper from KIIT University. Gopalda will be happy to interact with her. Gopalda already has her manuscript for last 2 months. I know he is the busiest person in Bhubaneswar now. I am still requesting him. We look for his advice, kind advice. We have found, now we wanted to see, we are expertise in cancer, we have expertise in virology. Let's them fight two bad things to fight for something, we would be benefited. And we found the similar things. Shomlata nicely worked on that, and our lab members also contributed that if we infect the cancerous cells, a metastatic cancerous cells with the virus, what happened? We found that magic things are happening, and which quite similar to COVID-induced anti-cancer activities. We found that they are replicating in a better way or higher way in the cancerous cells as compared to immune cells, because immune cells are the soldiers. They can resist the virus, but the cancerous cells, that is my philosophy. I do have to see these things technically or mechanistically. We have done good work, and we found that we know that cancerous cells are immortal in general, right? They are just like, in our mythology, we have heard about Raktobij, right? That they grow rigorously. So we found, and the target is anti-cancer drugs is target to induce apoptosis, to induce cell death to the cancerous cells. And that is a challenge without affecting the healthy cells. Just like an army's work is to find the terrorist or the enemies without hampering the civilians. We found that if we infect the cancerous cells with the virus, chikungunya, they induce apoptosis. They induce cell death, and they replicate higher. And they also restrict the immune suppressions mediated by the cancer cells by a specific immunosuppressive cytokine, that is interleukin 10. A detailed mechanism signaling pathways has been well explored by Shomlata. This is the work. You can see that IL-10 is the center, and we have worked on T cells, macrophage, everything together. And we are also working with immunosuppressive drugs, and couple of papers are there that we have used autoimmune disease drugs. It's basically immune modulations, but only they're interested, look for new models. We just play with T cells and macrophage. We can easily, this is bread and butter for our lab, we can easily modulate T cells and macrophage functions by activations or immune suppressions. And if we are getting any lead for natural sources or marine sources or plant sources, we can test whether they have the efficacy for T cells and macrophage modulation. And these are the papers. These are the ongoing work. Shoma is working, Partho is working, and Agniv is a new PhD student, and we have potential MSc students. We are working on trip channels and TLRs, and already the paper NRP1 is there for cancer things, but we are looking for Gopal Das comments. We are also looking for chikungunya virus, the same molecules for different platform. One is virus, another is cancer. And we are also with chemistry department in NISER. We are working with coral. It's a basically an inorganic compound with different porphyrin rings. We have nice lead for T cell modulations. It's a synergy between the School of Chemical Sciences and Biological Sciences. And these are the work. Another work, we are working on regulatory T cells, TLR, toll-like receptor, TRP channels, and immunomodulatory compounds on T cells and macrophage responses. This is our task, and we look for collaborations. This is our recent photographs, and we have thanks to collaborators, grant agencies, and the PhD students, and many of them are here. Thank you very much. I have finished in 17 minutes. Thank you.
Great.
A warm thank you to Dr. Chattopadhyay for his outstanding presentation. I had one question from Professor Kundus.
Dr. Subhasis, very nice way you explained in an elegant way.
Thank you, Dada. Yes, sir.
Melanoma induced immune suppression by infecting with the chikungunya virus. Have you checked that one in preclinical melanoma animal by developing the tumor in animal, or because melanoma metastasized to lung metastasis on in skin cancer?
True.
Have you developed that and shown that how it works?
Yeah. This is our future projects. In vitro we have tested, but the manuscript you have there, we have tested the melanoma mice model that NRP1 is regulating the chikungunya virus infections. Because of the biosafety issues, we have planned, because we have collaborations with ILS for animal model. In vitro we have tested that whatever the tumor microenvironments mimic that immunosuppressive conditions is regulated by the viral infections. Your specific question was about the animal model where you induce lung metastasis, whether in that case the viral infection reduce lung metastasis. Still that no, we didn't check it, but we are very much sure that it will do the similar things, because the molecular pathways, because melanoma things is established for last 50 years. Well established about all the mechanisms, everything is known, and we found in vitro all the mechanisms are shut down by the viral infections significantly, at least 60%. Shomlata can tell better, but you need to check for in vivo conditions.
So my suggestion will be, because the Institute of Life Science has an excellent experimental animal facility.
Yeah.
And my laboratory work in this field last more than 20 years. Then prove it, because that will go really towards the translational and the viral load and little bit targets and how it's working, mode of action.
Thank you, Dada. Thank you.
As a photographer, before we go for lunch,
I request Dr. Himanshu M. Padi, Chairman SGI and organizer of Nasi INSA conference, to please present the memento to Dr. Chattopadhyay, Associate Professor, NISER Bhubaneswar. I request Professor S.K. Acharya, sir, CEO of ITBI Sophitorium, to please present the potted plant to Dr. Chattopadhyay as a token of appreciation. Please.
Speaker, Professor Padi just showed me. So they have connected with YouTube. So all the speakers' presentation, 70 to 80 people from outside, they are just attending this meeting, and then they are observing that. Thank you, Professor Padi, for that. Yes.
I request all of you to come forward for a group photo.